Somewhere right now, in a laboratory you will never visit, a set of cells is dividing. Those cells belong to a woman who died in 1951. She was 31 years old. She weighed less than 50 kilograms. She has been dead for over 70 years. And yet her cells are alive, dividing, consuming nutrients, multiplying, in laboratories on every continent on Earth. The total mass of her cells produced since her death exceeds 20 metric tons. She never consented to this. She never knew it was happening. Her family did not know for 25 years. Her name was Henrietta Lacks. Her cells helped produce the polio vaccine, advanced cancer research, and validated the technology behind COVID-19 vaccines. She died in relative poverty at Johns Hopkins Hospital in Baltimore, Maryland. Her cellular legacy is one of the most consequential in the history of medicine. What follows is the story of how a dying woman accidentally became biologically immortal, and what that tells us about the nature of life itself. Henrietta Lacks was born on August 1, 1920, in Roanoke, Virginia, and raised in Clover, a small tobacco farming town, after her mother died in childbirth. She married David Lacks and moved to Baltimore, where they had 5 children. In January 1951, she noticed unusual vaginal bleeding and went to Johns Hopkins, one of the few hospitals in the region that treated black patients. Doctors found a malignant tumor on her cervix, an aggressive adenocarcinoma later confirmed to be driven by human papillomavirus strain HPV-18. On February 8, 1951, while Henrietta was receiving radium treatment, a tissue sample was taken from her tumor without her knowledge or explicit consent. The sample was sent to the laboratory of George Otto Gay, head of tissue culture research at Johns Hopkins. Gay had spent years trying to keep human cells alive outside the body. Every sample he had received, from cancer patients, healthy volunteers, other tumor biopsies, had died within days. Henrietta's cells were different. They not only survived, they thrived. Within 24 hours they had divided. Within a week they had doubled repeatedly. Gay's laboratory assistant Mary Kubicek was the first to observe the cells multiplying under a microscope. Gay named the line HeLa, the first 2 letters of the patient's first and last name, a code intended to protect her privacy. In the years that followed, he distributed HeLa cells to laboratories across the country and eventually the world, charging nothing, wanting only for science to advance. To understand why HeLa's survival was extraordinary, you need to understand why all the previous samples had died. In 1961, a cell biologist named Leonard Hayflick, working at the Wistar Institute in Philadelphia, made a discovery that overturned a foundational assumption of biology. For decades, scientists had believed that human cells in culture were essentially immortal, capable of dividing indefinitely as long as they were fed. This belief was largely based on work by the Nobel laureate Alexis Carrel, who in 1912 claimed to have kept chicken heart cells dividing for over 20 years. Hayflick was skeptical. Working with a cell line derived from human fetal lung tissue, designated WI38, he counted how many times the cells divided before stopping. The number was consistent, approximately 50 divisions. After roughly 50 divisions, the cells entered a state Hayflick called replicative senescence. They stopped dividing permanently, lingering in a metabolically active but reproductively inert state before eventually dying. Hayflick concluded that normal human cells carry a built-in division limit, a biological counter that counts down from birth. This is now called the Hayflick Limit. When Hayflick published his findings in 1961, the scientific establishment largely rejected them. The consensus built around Carrel's work was too entrenched. He was correct, and the field would spend 2 more decades catching up. The molecular mechanism behind the Hayflick limit was not identified until the 1970s and 1980s, through work that would eventually earn a Nobel Prize. At the ends of every chromosome sits a repetitive DNA sequence called a telomere. In humans, the telomere sequence is TTAGGG, repeated thousands of times. Telomeres act as protective caps on chromosomes, preventing them from fraying and from fusing with one another. Each time a cell divides, the DNA replication machinery cannot fully copy the chromosome's very end. As a result, the telomere shortens slightly with every division. After approximately 50 divisions, the telomeres are critically short. The cell detects what appears to be chromosomal damage and activates a cascade of molecular signals that permanently halt division. The telomere is the biological clock that underlies the Hayflick limit. In 2009, Elizabeth Blackburn, Carol Greider, and Jack Szostak received the Nobel Prize in Physiology or Medicine for this discovery. The enzyme that maintains telomeres is called telomerase. Telomerase is a ribonucleoprotein, a complex of RNA and protein, that adds TTAGGG sequences back onto chromosome ends after each division, effectively resetting the telomere clock. In most adult human cells, telomerase is silenced. It remains active only in stem cells, germline cells, eggs and sperm, and certain immune cells that need to divide extensively. This silencing is not accidental. It is a tumor suppression mechanism. A cell that can indefinitely rebuild its own telomeres is a cell that can divide without limit, which is the definition of cancer. HeLa cells are cancer cells, specifically cervical cancer cells transformed by HPV-18. HPV-18 is an oncogenic virus that causes cancer by integrating its own DNA into the host cell's chromosomes. When HPV-18 integrated into Henrietta Lacks' chromosomal DNA, it inserted its genetic code near regulatory regions controlling telomerase expression. The The viral proteins E6 and E7, encoded by HPV, simultaneously inactivated two of the cell's most critical tumor suppressors, p53 and the retinoblastoma protein, RB. p53 is often called the guardian of the genome. It detects DNA damage and either triggers repair or forces the cell to self-destruct. RB is the molecular brake that prevents cells from entering the division cycle. With both p53 and RB disabled, HeLa cells lost the ability to detect damage, enforce senescence, or initiate programmed death. Simultaneously, the HPV integration caused telomerase to be permanently and massively overexpressed. HeLa cells produce telomerase continuously in every cell cycle. Their telomeres never shorten to a critical length. They cannot senesce. They cannot die of replicative exhaustion. Within the constraints of available nutrients in space, they are biologically immortal. In 1952, the United States faced the worst polio epidemic in its history. Nearly 58,000 cases, over 3,000 deaths, and tens of thousands of children left paralyzed. Jonas Salk at the University of Pittsburgh was racing to develop a vaccine, and he needed a way to grow large quantities of poliovirus for testing. HeLa cells were susceptible to poliovirus infection, making them ideal culture vessels. The National Foundation for Infantile Paralysis funded a dedicated HeLa production facility at the Tuskegee Institute in Alabama. By 1953, the facility was producing 20,000 tubes of HeLa cells per week to supply Salk's vaccine trials. The polio vaccine, declared effective in 1955, one of the greatest public health achievements in history, was developed using Henrietta Lacks' cells. By the mid-1960s, HeLa had been sent to virtually every major research laboratory in the world. And then, scientists began to uncover a disturbing problem. In 1966, geneticist Stanley Gartler presented a paper at a research conference in Bedford, Maryland, that stunned the assembled scientists. Gartler had analyzed 18 of the most widely used human cell lines in existence, from different laboratories, different patients, and different tissue types. Using a genetic marker specific to people of African descent, a variant of the enzyme glucose 6-phosphate dehydrogenase, he showed they were genetically identical. They were all HeLa. HeLa cells, faster dividing and more aggressive than nearly any other cell line, had contaminated culture after culture through airborne droplets, shared equipment, and careless handling. Estimates suggest that by the 1970s, between 10 and 20% of all cell cultures in use globally had been secretly replaced by Henrietta Lacks' cells. Decades of published research had been conducted on HeLa, while researchers believed they were working with something else entirely. HeLa cells were the first human cells sent into space, aboard a Soviet satellite in 1960, to study the effects of weightlessness on human biology. More than 110,000 scientific papers have cited HeLa cells as a research tool. The mRNA technology underlying the Pfizer and Moderna COVID-19 vaccines was partially validated using HeLa cells. The total mass of HeLa cells produced since 1951 exceeds 20 metric tons, thousands of times Henrietta's own body weight, still growing. Henrietta Lacks died on October 4, 1951, 8 months after her first visit to Johns Hopkins. Her family did not learn that her cells had been taken until 1973, when a researcher contacted them seeking blood samples for genetic mapping. For decades, the Lacks family had no legal rights over the cells, received no compensation from the multi-million-dollar industry that had been built on HeLa, and struggled to afford the health insurance that those cells had helped make possible for millions of others. In 2010, journalist Rebecca Skloot published The Immortal Life of Henrietta Lacks, bringing the story to mainstream public attention for the first time. The book spent more than 75 weeks on the New York Times bestseller list. In 2013, following the publication of the full HeLa genome by European researchers without family consent, the National Institutes of Health negotiated a landmark agreement with the Lacks family. Researchers wishing to access HeLa genomic data would need to apply to a committee that included 2 members of the Lacks family, the first time any family had been given formal governance over a cell line derived from their relative. It did not include financial compensation. The case of Henrietta Lacks became the defining example in the global debate over who owns human biological material, and whether consent obtained under the medical norms of a racially segregated hospital in 1951 constitutes meaningful consent at all. Here is what Henrietta Lacks' story forces you to consider. Right now, in the cells of your body, telomeres are shortening with every division. The cells in your skin, your gut lining, your immune system, each one is counting down. You have a finite number of divisions left in most of the cells you depend on. That finitude is not a flaw. It is the same mechanism that prevents your own cells from becoming HeLa. The Hayflick limit is the price of not being a tumor. Henrietta Lacks' cells escaped that limit because they had no choice. A virus had dismantled the architecture of cellular death from the inside. And those cells have not stopped. They do not have an experience of time or meaning. They are a biological process that has lost its off switch. But they have done something no other cells in history have done at this scale. They have outlived their source by more than 70 years. They helped end polio. They built COVID vaccines. They reshaped our entire understanding of how cells age and die. In 1951, a black woman in a racially segregated American hospital had a piece of her tissue removed without her knowledge. That tissue is now woven into the fabric of modern medicine. Her name is Henrietta Lacks, not HeLa, not a cell line identifier. A woman, a mother, a farmer's granddaughter from Clover, Virginia, who died at 31 years old and has never stopped contributing to the world. The cells do not know they were hers. But we do, and that may be the most human thing about all of this.